Today, we're diving into one of the biggest questions there is. How does life, well, build itself? We're going to go on a journey from a single, simple molecule all the way up to a living, functioning cell to figure out how it all comes together from the ground up.
Okay, so take a look at this plant cell. You know, it's so easy to just picture a cell as this tiny little bag of chemicals, right? But that's not it at all. The reality is, wow, it's way more spectacular. Think of it like a bustling, super organized city. And this isn't random chaos, far from it. Every single part has a job, a place to be. Its movements are all tightly controlled. It's like this perfectly timed symphony happening on a microscopic scale.
Which, of course, brings us to the big question, the one we're tackling today. How does nature pull this off? I mean, how do you start with just simple, non-living chemicals and end up with something this complex, this dynamic? How do you go from just a random pile of parts to a fully functioning factory?
So where do we even start to answer a question that big? Well, the secret, the key insight from biochemistry, is that life isn't just poof, built all at once. Nope, it's assembled in these really clear stages, a 4-level structural hierarchy. And this hierarchy, that's going to be our roadmap for this whole journey.
And here it is, our 4-step plan. First, we start at the absolute bottom with the simplest building blocks. We call these monomeric units. Then we'll see how those little guys get linked up into these long chains called macromolecules. After that, we'll see how those chains assemble themselves into the actual working machinery of the cell, the super macromolecular complexes, and then, for the grand finale, we'll see how all of that machinery comes together to create the whole factory of life, the cell itself.
All right, let's dive in. We're starting at the bottom, tackling those first 2 levels. And honestly, the best way to think about this is to think about language, right? You can't write a word without an alphabet. Well, biology works on the very same principle.
And what's just wild is how small life's alphabet actually is. I mean, think of all the chemical elements out there. Biology is super picky. It only uses a tiny select group of these monomeric units. You've got your amino acids, which build proteins. You have nucleotides, which build DNA. And then you've got sugars and lipids for structure and energy. That's pretty much it. These are the fundamental letters that spell out almost every living thing you can imagine.
Okay, so you have the letters. How do you turn them into actual words? Long, stable things like a protein or a strand of DNA. Simple. You need glue. And in the world of biochemistry, the main glue is something called a covalent bond. Seriously, just think of it as molecular superglue. It is incredibly strong, and it forges these permanent links that you need to build those long, tough chains we call macromolecules.
Okay, so now we've made our words, these long chains of macromolecules. But, a random list of words doesn't make a story, does it? The next step, level 3, is all about taking those words and arranging them into sentences, into functional structures. We're talking about the complex machines that actually do all the work inside the.
And this is where things get really clever. If those covalent bonds were the superglue, the bonds that assemble these machines, well, they're more like Velcro. They're called non-covalent bonds. And here's the cool part. On their own, each little hook and look is way, way weaker than the superglue. But that weakness is actually their greatest strength. Because they're flexible and reversible, it means the cell's machines can be built, taken apart, and rebuilt over and over again whenever they're needed.
But wait a minute, how can this weak Velcro possibly hold together these massive cellular machines? The answer is simple: strength in numbers. A single one of these interactions, yeah, it's temporary, it's weak. But when you get hundreds or even thousands of them working together between 2 big molecules, suddenly you've got a super stable and very specific connection. That right there is the secret sauce for building things that are both complex and incredibly dynamic.
Okay, so we've got the hierarchy down. Building blocks, chains, machines, factory. But so what? Why does this actually matter? Well, it's a huge deal for scientists because it points to a massive challenge they face: the world of difference between studying something in a nice clean test tube and studying it inside a real, live, messy cell. And this is like a central problem in biochemistry. For decades, scientists studied molecules in vitro. That just means in glass, like in a test tube. It's clean, it's simple, you can control everything. But the inside of a cell, in vivo, or in the living, is a whole different ballgame. It is a crowded, sticky, gel-like mosh pit where a molecule is constantly crashing into thousands of other things. And all those interactions, they can completely change how that molecule acts. And that's where you see its true biological purpose.
So here's the big takeaway. If we really truly want to understand life, We can't just look at the parts in isolation. We have to study them in their natural habitat. We've got to appreciate how this entire hierarchy, from those tiny building blocks all the way up to the complex machines, functions as a whole, right there in the middle of all that organized chaos inside a living cell.
And that brings us to one last, really powerful idea. It's captured perfectly in this famous quote by the biochemist Jacques Monod. Everything we've talked about today, this step-by-step construction plan, the superglue and the Velcro, the tiny little alphabet of life, it's not just true for one thing. It's universal. These rules apply to the simplest little bacterium just as much as they apply to the most complex animal. This is the shared chemical blueprint that connects every single living thing on this planet.